Hi, we're the eSeers. My name is Emily.
My name is Sinclair.
I'm Ella.
I'm Eric.
And I'm Rishi, and today we're going to cover metagenomics and studying the human gut health. Now, there are thousands of species living in our intestine right now. In our intestine, there's a gut microbial community. We'll get over the definition of microbes sooner or later, but this community is really important when it comes to regulating immunity and regulating metabolic processes. Fun fact, it's really helpful that it can even be considered an endocrine organ. Typically, human gut microbiota behaves kind of like a multicellular organism. It consists of 200 bacterial species. To learn more about this human gut, we're trying to understand how microbes work and their specific roles. And in order to do this, we're going to use metagenomics. Metagenomics was discovered in 1998 by Handelson and Rodon and serves a huge purpose in studying our gut microbial community. So, as you can see, the definition of metagenomics is a study of collection of genetic material from a mixed community of organisms. If there are two bacteria living together and you take the DNA sequence, you'd get both bacteria's DNA because metagenomics takes a community of multi-organisms. So, materials to get from metagenomics can be retrieved to our gut, soil, water, many different things. And what we can use with metagenomics is kind of find out the diversity and dysbiosis that will be covered later in the gut microbial community. And overall, we're trying to understand the relationship between health and disease, identifying novel genes, etc. So now Eric will discuss the two different types of metagenomic study of the gut microbe.
Yeah, so there are two main types of metagenomic approaches when we're studying the human gut microbiome. They are the descriptive metagenomic and functional metagenomic approaches. And just to give a brief explanation of the difference between the two, descriptive metagenomics can be used to sort of reveal like the dynamics in population and like community structure of the microbiome in our gut, different environmental conditions, and so on. And functional metagenomics is much more about the relationship and interaction between the gut microbiome and the host, and between each microbe and other microbes. So this can help enlighten us about the exact functions of each microbe group or microbial species within the gut microbiome. Next slide, please. So within descriptive metagenomics, we have two different types of analysis, and that's genomic analysis, which covers the genes between different microbes, which can help sort of also look at the population and structure of each biome, as stated before, and also spatial distribution analysis, which, as you can see in this diagram, can help us also look at different distributions and of species and analyze the population as a whole. Next slide. But what's mainly focused on within the human gut biome is functional metagenomic study. And there are a multitude of extremely interesting applications and frontier pushing ideas within this discipline. So one is using E. coli to sort of discover gene expressions beyond the genome sequencing that we did in our class, like the shotgun sequencing, where we sort of split RNA into a bunch of different small sequences and then sequence that. There is this expression called translational fusion, which is unable to be found, like inaccessible by shotgun sequencing. And what scientists did is they used E. coli hosts to show presence of that expression, which was impossible using previous methods. So that's an interesting application of functional metagenomics. Another one would be sort of environmental uniquities within different populations, not just in bacteria, but within humans and their gut microbiomes. So an example would be in Japan, there is a higher percentage of human gut microbiomes that include the enzymes porphyrinase and agarase. And the explanation for this is that Japanese eat seaweed regularly. And this is an example of like lateral gene transfers. So that was found by functional metagenomics study. And finally, and I think the most important application is the discovery of biosynthesis, synthetic pathways. So a lot of the sort of microbes in our belly are also responsible for providing essential enzymes and metabolites to our daily function. And well, one example would be carbohydrate active enzymes or CASIMs, which were found in the metagenome using functional metagenomics. And yeah, they are essential to our function to catabolize fibers and are extremely important to the human gut microbiome. Next slide. So in the same way, we can see how these bacteria contribute to our health. Bacteria can help digestion. They break down fibers to like short chain fatty acids. They also synthesize, like previously mentioned, essential amino acids and vitamins in addition to those enzymes. And one example I didn't mention was butyrate, which a scientist, Vital, researched using this genome sequencing and was able to find out which bacteria were responsible for that production. And finally, bacteria also play a role against disease. They can protect against pathogen attacks. All these factors combined demonstrate bacteria's importance in keeping us healthy and how metagenomics can help us research and clarify their roles.
Metagenomics and gut health. There were two very notable projects that helped with intestinal biome research. They helped facilitate these studies through studying fecal samples. So there was MetaHit from Europe and the Human Biome Project from America. Results concluded from these two studies that there are about 160 bacterial species in each individual that remain relatively stable in the long term. The gut's population was, from this research, we were able to determine that the gut has three main types of organisms called enterotypes that make up the microbial population of the gut: Provitella, Ruminococcus, and bactericides. Every individual's composition is pretty unique for the percentages of each type of enterotype. The composition and diversity of the gut's microbiome can be influenced by many factors, including diet, age, geography, drugs used like pharmaceuticals, and environmental substances. What exactly is dysbiosis? Dysbiosis is an imbalance of in the microbial composition of the gut. As you might be able to imagine, an imbalance of microbes can be extremely harmful to the health of the gut and often interfere with the stomach's ability to digest food properly. Dysbiosis is associated with many diseases such as diabetes, IBS, colorectal cancer, colon polyps, malnutrition, and many more. Luckily, metagenomics can help identify cases of dysbiosis in individuals. It can also be used to help study interactions between the intestinal bacteria in an individual and between their body. For example, intestinal bacteria can sometimes help mucus secretion in the gut.
One of the roles of metagenomics in the study of human gut is to sequence bacterial DNA. Many sequencing projects have taken tens of thousands of gut samples to try to classify and learn more about gut bacteria. This allows scientists to identify new bacteria, increasing what we know about the microbiome. Sequencing also reveals insight about the function of the bacteria and its role in our health. Despite extensive efforts, what we know is not a complete collection of all gut bacteria. This becomes another problem when lots of bacteria collections are not sampled from understudied populations, making what we know quite skewed. Luckily, sampling more bacteria from more diverse sources can help reduce the issue. Why haven't we sequenced all gut bacteria? One reason is misrepresentation. Lots of microbiomes remain unsampled from understudied populations like Africa. Also, lots of gut sequences have been taken from patients, which means that we know more about microbiomes corresponding to medical conditions and relatively less about healthy microbiomes than in the proportions that they naturally show up in. Also, some bacteria are simply less abundant. Even if some types of bacteria aren't as prevalent in the microbiome, they are still likely to perform important roles. It might surprise you, but the gut microbiota is a critical controller in brain aging, cognitive impairment, and just aging in general. Over 100 years ago, Elie Metchnikoff linked the gut microbiota with human health and aging for the first time, theorizing that health and lifespan could be enhanced and prolonged by regulating the gut microbiome via the consumption of host-friendly bacteria found in yogurt. The gut microbiome probably starts its development before the baby is born, and over the years it undergoes many changes affected by things like your diet, gender, and even the way you were born. In an elderly person, the biodiversity decreases, which is associated with cognitive decline. Metagenomics is what allows scientists to identify fluctuations in what is normal in microbiomes in different ages. Clinical evidence has connected the gut microbiota dysbiosis with cognitive deficit in the elderly, as subjects with cognitive decline and brain amyloidosis have lower abundance of anti-inflammatory erectile and higher abundance of pro-inflammatory Escherichia or Shigella compared with that of control subjects. Other strands of bacteria have also been correlated with cognitive decline and brain diseases. Identifying microbiomes associated with aging can possibly help scientists better mitigate the effects of aging.
And continuing on, we will look at findings having to do with the most recent pandemic. Utilizing metagenomics to study gut health has helped researchers make correlations between gut health and microbiome diversity and many diseases. Most recently, the COVID-19 pandemic has left scientists to scramble for research. However, many have found that applying metagenomics to studying human gut health could be a helpful way to combat COVID-19, or at the very least, learn more about it. Now, again, this isn't really necessarily a new approach. Previous research on both humans and mice has seen correlations between gut dysbiosis and other diseases, so it was reasonable to study COVID's effects on our gut microbiome health. Scientists would study the actual gut microbiome and use software such as BLAST to map the reads of data. It was found that imbalances in microbes in our guts caused by the COVID infection may bring about microbial dysbiosis. Some of the first research done on this topic was in Hong Kong, where scientists studied the feces of hospitalized patients diagnosed with COVID-19. It seemed to be a running trend in which most of these research studies were to examine the gut microbiome content present in those with COVID and compare them to those of control or healthy individuals. They found many interesting things. First of all is this bacteria in which I have no hope of pronouncing. The bacteria in italics is known for promoting gut health, so a distinct lack of it might contribute to the evident dysbiosis. It has also been correlated to the severity of the COVID case. Interestingly enough, they've also found an increase in opportunistic microorganisms. A quote from the article stated that researchers smartly pointed to the microbiome as a key element in understanding the etiology, infection, and transmittability processes of the emerging SARS-CoV-2 virus. So we may see that even though we still need many advances in computational science, investigating gut health could one day give us a good look at other emerging or existing diseases.
So even though metagenomics may have its limitations, as you know, there's many benefits to doing so. As we can say, diversity in the gut, revealing new genes, discovering new bacteria in the gut, and figuring out more about their functions. And there's just so much we can do, especially when it comes to using it for even COVID-19, something that's impacted all of us, right? Even bioactive compounds, drugs from marine microbes, new genetic information on enzymes, making antibiotics. There's so many things we can do with metagenomics. So overall, it is very powerful. And here are some of our sources that we have. And of course, more sources continued. But overall, hope you guys enjoyed our presentation. And thank you all for listening.